All right, guys, so here we are, our next section for our EOC review. This is part 2. This one's going to be all about genetics. We're going to get into the classification of living things and then origin of life and evolution. So going back to genetics, we have that monk, Gregor Mendel, who studied the traits of pea plants, and he is the one who actually came up with traits or how traits were passed down from generation to generation. Now, he concluded that each organism had two factors that controlled each of its traits. We call those factors genes, and they're located on chromosomes, and you get two of them, one from mom and one from dad. Now, genes exist in alternative forms. The different forms of a gene are called alleles. Remember, like you might have inherited a blue-eyed gene from dad and a brown-eyed gene from mom, for the eye color gene. Those different forms are the alleles. Now, Mendel, when he was working with the pea plants, observed traits that were dominant and traits that disappeared, which he called recessive. Dominant traits we write with a capital letter, recessive traits we write with a lowercase letter. Now, here's a little bit of vocabulary to go along with our section on genetics. The physical traits that you display, those are considered your phenotype. The alleles, the copies of the genes that you have, that's called your genotype. So genotype, what genes do you have? Phenotype, if you just think physical characteristic or physical traits. And sometimes your genotype can't always be known by your phenotype, okay? Because you might look a little bit different because of those recessive genes. Now, an organism is homozygous for a trait if the two alleles are the same. So if I had a brown-eyed gene and a brown-eyed gene, I'm homozygous. Heterozygous for a trait is if you have two different alleles. Okay, so a blue-eyed gene and a brown-eyed gene. Now, Mendel came up with this law of segregation, states that every individual has two alleles of each gene, and when the gametes are produced, the gametes only receive one copy of those alleles. And we know that because we talked about meiosis last week and how we start with 46 total chromosomes, but that chromosome number gets in half because each sperm and egg cell only gets half the amount of chromosomes. Now we have this thing called a monohybrid cross. If you remember correctly, we go through, we determine what alleles that the parents have. First one, we're going to say heterozygous tall parent. So if they're heterozygous, that means they have two different forms of the gene, a big T and a little t. And then again, when we talk about this, that big T is dominant, so that parent is going to be tall because like the short gene is covered up. The other parent in this instance is homozygous short genes. Homozygous, two of the same alleles. If they're short, they're going to have two small t's. For my next example, we have both parents are heterozygous, so you're going to go ahead and fill that in. When you go and fill it in, you should have ended up with answers that look like this. If you don't remember how to do those, please message me and I can send you a video that goes through it a little bit more clearly. Okay, the next thing, Mendel came up with a law of independent assortment. It states that genes for different traits, for example, seed shape, seed color, are inherited independently of each other. So if you have a parent pea plant, the genes for shape and color will be inherited independently for each other. And we can do that by doing a dihybrid cross. Okay. So the dihybrid cross, we go through and we do the FOIL method to determine what are the genotypes of the parents. Then we go through and we list the genotypes along the top and the sides for both parents. In this one, I said both parents have the genotype big R, big Y, little y. So we need to figure out what would be in each of the sperm and the egg cells and then fill that in. So our gametes for the dihybrid cross, once we've done that FOIL method, which I've gone through before and I'll review with those of you in class with me, we would have big R and big Y, big R, little y, big R, or little r, big Y, and then little r, little y. We place those on the top and the sides, and then we go and bring them down and across to meet each other, just like we do with a regular Punnett square. This is the dihybrid cross. Remember, anytime we have heterozygous for both traits, we're going to have that 9:3 to 3:1 ratio. Now we have to talk about things called sex-linked genes, because the X and the Y genes determine the sex. Genes located on them show a pattern of inheritance called sex linkage. So sex linked, they're on the X or the Y chromosomes. Remember, X and X are female, X and Y are male. And females can only pass down the X to their offspring, and males can pass either the X or the Y chromosome. In order for a recessive sex-linked gene to be expressed in a female, she must have the gene on both copies of her X chromosome. Otherwise, she's going to have a dominant one to cover it up. So we've got our dominant alleles. Okay, here's our father. You can see that he's shaded blue, which means he's unaffected. He does not carry that, well, actually not carry, but does not display the sex-linked characteristic. Then we have our female. She is a carrier. She has one copy of that disorder. When they pass those down, if we go through and we actually do a Punnett square, you can see we would end up with a 25% chance of having a male born with that disorder. We would have one female born with normal chromosomes on both sides, normal genes. We'd have another son born with normal one, and then one female that's going to be a carrier just like her mother. And we have this thing called incomplete dominance. And incomplete dominance this is cases where one allele is not completely dominant over another. Like if we breed this red flower with this white flower, our offspring are going to be pink. So we'd end up with a blending of those characteristics. Then we have codominance. In codominance, the phenotypes are both expressed equally. Blood type is a codominant trait. It also exhibits multiple alleles, which means that there's more than two different versions of the gene. Like in blood types, you can have type A, you can have type B, you can have type O, and you can order and then AB, and you can have any combination of genes because there's multiple alleles. So anyway, with blood typing, if you go through and do Punnett squares, you can see if you have type A and type B, you inherit both copies, you're going to be co-dominant. You're going to express both of those equally. Okay? Okay, the next thing, pedigrees. If we look at pedigrees, again, just to refresh your memory, it shows the patterns of inheritance from generation to generation. Squares represent males, circles represent females. Shaded means they're affected by a disease, and unshaded means they're not affected by the disease. So we've got this pedigree here. We have our affected male up here in the top, number 1, unaffected female, number 2. Then we've got our next row here. We have one affected male, normal versions on the female, normal versions on this male, and so on. Okay, and so it says what genotype, what would be the genotype for individual number 6 on this pedigree. So if we look at him, he is affected by this trait. Now it's a male. Is he going to have X and X or X and Y? Well, male, X and Y, and he's affected by the trait. So is he going to have the recessive version, which means he will have the genetic disorder, or the dominant version, which means he does not have it? Well, hopefully you've picked D, because he has the disorder, he's shaded, so he should be X with a little h next to it, Y. Moving on from there, we started to talk about natural selection and evolution. Charles Darwin developed a scientific theory of biological evolution that explains how modern organisms evolved over long periods of time through descent from common ancestors, and he called this theory natural selection. It's basically survival of the fittest. So some conditions for natural selection, you have to have competition. It means that we all struggle to survive, right? And in the wild, you want to obtain food, you want to have space, you want to have mates, all of these different things, and you're always in competition with the same species and with other species. And then you have variation and adaptation. So natural variations, inheritable traits, may allow one individual to survive better than another, allowing it to reproduce and pass on its traits. And then survival of the fittest. So fitness describes how well an organism can survive and reproduce in its environment, and more fit organisms are more likely to reproduce and pass on their genes, thus changing them if they have an adaptation or a variation. So it will change the gene pool over a long period of time. Over time, series of chance occurrences can cause an allele to become more or less common in a population. And this random change is called genetic drift. We can have this thing called the bottleneck effect, which is the reduction of that allele frequency, so how many different versions of the gene due to disease or disorder. Then we can also have a thing called the founder effect, which means that we might have a group that emigrated or immigrated in or out of an area, and that can also change the alleles of the versions of the genes found in that gene pool. Also, we have genetic variation, and most differences come from crossing over. Remember when we talked about meiosis? Told you crossing over is really important. That's when the genes flip-flop spots. And then also independent assortment. We have different versions of the genes ending up in different sperm and egg cells. A few differences come from random mutation, and things increase, both these things increase the genetic diversity among organisms of the same species. Okay, so we're going to change gears and talk a little bit about classification. Here is our different levels of classification. We have domain, kingdom, phylum, class, order, family, genus, species. We also talked about these things called cladograms. Cladogram shows the links between groups of organisms showing evolutionary lines or lineages branched off from common ancestors. So way back here at A, we have some kind of common ancestor. Over time, they developed some different traits. Okay, so these ones branched off. All of these ones after this have some of the same features. Okay, so we're traveling somewhere here. Okay, we would have had another node here of something else changing and evolving where they developed some kind of trait, and these ones branched off into their own families and groups. Okay, whereas all of these down here are going to have some kind of feature that's common to all of them as well. I'm going to have you go through the classification chart. We actually copied this in our notes already, but take a look at that. You have a copy of that in your notes because we copied a classification chart. Please, please, please study it. Okay, then we have the origins of life on Earth. Scientists hypothesize that life started by some kind of chemical process where simple organic molecules must have formed. Those molecules somehow became organized into more complex molecules, beginning proteins and carbohydrates and nucleic acids, okay, and fatty acids, lipids. And RNA was thought to be the first genetic information in these cells. So the first forms of life may have been prokaryotic forms that evolved from RNA carrying the genetic information. And because Earth's atmosphere lacked oxygen, scientists propose that these organisms were more likely anaerobic, which means that they do not require oxygen and could survive in really extreme environments, kind of like our archaebacteria. If you look at that classification chart, we had that archaebacteria, they're extremists. They live in super hot or no oxygen or sulfur springs, all sorts of really crazy extreme environments. Then we have this Miller-Urey experiment. This actually tested the question, could organic molecules come together and form under the same conditions on early Earth? So if you look at their experiment, they suggest how mixtures of organic compounds necessary for life could have arisen from similar compounds. And so they had things set up like to represent Earth's primitive oceans. They had electrodes put in there to represent the lightning that filled our atmosphere at the time. One vial that was filled with the gas. And so they were boiling because remember, the oceans were boiling with volcanic eruptions and all sorts of different things. And so these boiling things would evaporate, go into the atmosphere where this electricity would shock them and give them some energy to start some kind of chemical reaction. And then once this happens, this collecting trap down here, after 7 days, they checked it and they found that they actually had made amino acids, which are the building blocks of proteins. Okay, our next theory for how life started is called the endosymbiotic theory. And it proposed that eukaryotes actually evolved through a symbiotic relationship between ancient prokaryotes. Remember, symbiotic relationship is organisms living in a very, very close relationship with each other. And so what they suggest is that a prokaryote at some time ingested some kind of bacteria. Remember, prokaryotes are bacteria. So one of them ate up another one, okay? The ones that they ate up were protected by, for some reason, they were living inside of them and actually helped to produce energy for that other bacteria. And over a long period of time, those new bacteria became mitochondria. They were no longer able to live on their own because they lived inside that bacteria for so long. And so some primitive prokaryotes also ingested these other kinds of bacteria which contain photosynthetic pigments. And then those also no longer be able to survive on their own after long periods of time and became chloroplasts. So for evolution to actually happen, it's very small changes over long periods of time due to natural selection. Some evidence for evolution is fossil record comparing the anatomy of organisms, embryonic development, and then DNA evidence. We talk about some anatomy. Structural features with common evolutionary origin are called homologous structures. So if you look at the bone structures here, we have our finger bones, we have our wrist bones, the two bottom arm bones, and then the top one upper arm bone. We can call these homologous structures because they're similar in their arrangement and their function, or both. We also have things that are called analogous structures. These actually do not have a common evolutionary origin, but they're similar in their function, like dragonfly wings and birds' wings. They are not related species, okay? They evolved separately. But because wings was a good adaptation, they did evolve these similar features. Another type of body feature that suggests an evolutionary relationship is a thing called a vestigial structure. This is a body structure that is in that present day organism no longer serves its original purpose, but was useful in an ancestor. One example are little tiny leg bones still found in the skeletons of whales. At some period of time, they had legs because they were on land. They evolved to go back into the ocean where these legs just shrunk down and shrunk down and shrunk down because they no longer needed them.